Welcome to our lesson on the uses of glucose. The key questions for today are 1. How is glucose produced in photosynthesis used by the plant? 2. How is glucose stored in the plant? 3. What other molecules can glucose be made into? 4. What other nutrients do plants require? 5. How do you test for the presence of glucose and starch? The key words you will need to know are glucose, starch, respiration, lipid, synthesis. Plants produce glucose through a chemical reaction called photosynthesis. This occurs in the leaves of green plants where the cells are specially adapted. Glucose is a type of simple sugar and has the chemical formula C6H12O6. Plants produce a surplus of glucose, so not all of the glucose produced is needed for respiration. The glucose can be stored in a plant as starch, which is a more complex, insoluble carbohydrate. It is important that the starch is insoluble, as this means it does not affect the water potential of the cell it is stored in. This can mean that osmosis will not occur, as starch is not a solute, as it does not dissolve. This means that starch can be stored in a plant until it is required. When the plant needs it, it can be broken back down into glucose and used. Plants also use glucose to make cellulose. This is the strong material that cell walls are made out of. Plants can strengthen their cell walls by converting glucose into cellulose. Glucose can also be made into other biological molecules, including lipids such as fats or oils and proteins. Proteins are synthesized using amino acids that can be made using glucose and nitrates that are taken in from the soil through the roots of the plant. Task: where in a plant is glucose produced? Extension: why must glucose be stored as starch inside plants? Glucose is produced in the leaves of the plant. It is then transported through the phloem to different parts of the plant. If the glucose is stored, it is converted to starch because starch is insoluble and will not cause the cells to take on water by osmosis. Glucose and starch can be identified in a plant using simple chemical tests. To test for reducing sugars, such as glucose, the sample must be crushed up and added to Benedict's solution. The mixture should then be heated, and if glucose is present, a colour change from blue to green, orange or brick red will occur. Brick red indicates a large amount of glucose present, orange some glucose present, and green a small amount of glucose is present. To test for starch in a sample, the sample must first be bleached and the cell walls will need to be broken down. For a leaf sample, it should be boiled in ethanol and then lay onto a white tile. Iodine is then used to test for the presence of starch. A drop of iodine will turn from orange to blue-black if starch is present. Task: a plant with variegated, two-coloured leaves was left in sunlight for several hours. Pieces of one of its leaves were then detached, removed, and tested for sugar. The diagram below shows the results. Explain as fully as you can why the yellow region of the leaf had not produced sugar. The yellow part of the leaf does not contain the green pigment chlorophyll, which absorbs the sunlight and allows photosynthesis to occur. So, no sugar would be produced in the yellow part, as no photosynthesis would take place. Extension. What would you expect to see if a green part of the leaf was tested using Benedict's solution? When tested with Benedict's solution, we would expect to see the green part of the leaf turn the solution from blue to green, orange or brick red to indicate the presence of glucose. This glucose would have been produced during photosynthesis.